Very good morning to everyone.
Good morning, Dr. Shilpa. Okay, welcome everyone to this one-day symposium focused on 3D culture. As we all know that primary cell culture is going through a massive transformation with the 3D cultures now coming faster than we would have even believed till about a few years ago. Uh, the uh, the coming up of 3D culture, organoids, and technologies like this are primarily very, very important and useful for our goal of getting into artificial organs, of goal into getting into biomedical printing, because that is where the next frontier of healthcare, as we can call it, is perhaps going to be. My name is Vipul Jain. I'm the founder and CEO of the Advanced Cells Group. And under the Advanced Cells Group, we have a branch called Kosheeka, where we produce on-demand primary cell cultures, both 2D cultures and 3D cultures. Our ultimate aim, again, is to be in the forefront of innovation when it comes to biomedical printing, 3D organ printing, and creation of working models of organs to assist both pharma companies, CROs, research models, different research models, etc. I welcome you all on behalf of the Advanced Health Group to this symposium. We have an extraordinary lineup of speakers today. To begin with, we'll have Dr. Suresh PK from VIT Vellore, the School of Biosciences and Technology. And he's going to be talking about how culture is shifting from 2D to 3D and how 3D culture is beneficial for drug discovery and cancer research. Up next would be Professor Tully Day from the Pune University Institute of Bioinformatics and Biotechnology. Dr. Day is going to talk about how 3D cultures is going to help in creating a natural environment for cells to do our research. Dr. Sivarajan T from the 𝐊𝐭𝐞𝐞𝐤𝐚 group in Bangalore is then going to be talking about organoids from a more industry point of view, how organoids are extremely important and what exactly they do. Then it will be the turn of Dr. Christophe Chesnay from vPredict in France, the makers of the HEPA-RG cell line. Dr. Chesnay will be talking about how 3D culture can help in bioartificial organs with a focus primarily on the use of 3D culture in HEPA-RG and bioartificial liver. And to top it all off, we'll have Professor Falguni Pati from IIT Hyderabad, who's going to be talking about the 3D bioprinting for medical applications. So we can promise you it's going to be a wonderful, wonderful day of learning and understanding how the future is going to pan out for primary cell cultures and the shift towards the 3D cultures. I'm now going to introduce Dr. Sachin Kadam, the Chief Technology Officer of the Advanced Cells Group and the host and moderator of this symposium. Dr. Kadam is going to be your moderator and will guide the entire symposium through the entire day. Dr. Kadam, your baby now, please take over.
Thank you so much, Vipul. Indeed, it was a great introduction about Kosheeka and Advances. So good morning, friends. So you all know what Advances Group do and what Kosheeka is. So I would just like to top it up on that the Kosheeka's main USP is that we prepare the primary cell culture cells as per your requirement and your demand. So whatever your request is, your demand is, you can just contact us and maybe we can get back to you with solution providing the primary cell culture for your use. So let's start with today's symposium. Today, 3D culture has gained increased momentum in biotechnology research and pharmaceutical industry. Over last few years, we have seen that 3D culture market is growing, at enormous rate. Like the predicted value was 3.2 billion by 2027. And researchers there globally, they have started exploring the potential of 3D culture and its in vitro application for drug discovery, validation, or then safety, toxicity. So as cell biologists shifting their focus from 2D to 3D, it is important for us to know the recent development in 3D cell culture technologies and their application. This is the main reason behind organizing this symposium, where we will inviting eminent speakers, experts in various 3D technology branches to make us aware about the same. So let's start with today's symposium. I would like to invite Professor P.K. Suresh. It's my actually really a great pleasure and honor to introduce a great academician and a researcher with vast experience of more than 20 years in the field of pharmacology, toxicology. So please welcome Professor P.K. Suresh, who's working as a professor higher academic grade in School of Biosciences and Technology, Vellore Institute of Technology, VIT, Vellore. Professor Suresh received his doctoral degree in environmental health from University of Cincinnati College of Medicine. He has mentored several doctoral and postgraduate students until now. With many government-funded projects to his credential, he is also a participant or recipient of Science and Engineering Research Board Travel Fellowship in 2014 to chair the session in Beijing. Currently, he is also a principal investigator on SERB project on drug discovery, drug delivery. His main research area is in cell culture and drug delivery related aspects and their clinical side, with special emphasis on drug development model. Today, he will be speaking on transition of cell culture over the last few years from 2-dimension to 3-dimension. The exact title of his talk is 2D Cell Culture System for Natural Molecules, Drug Developments and Delivery System. Transition of 3D system, focus on certain hallmarks of cancer. So without any further delay, I invite Professor Suresh. Sir, screen is now open for you. You may share your screen.
Yes, thank you for the wonderful introduction. I want to thank the organizers for giving me this opportunity to present today. Screen visible? Is the screen visible?
Not yet, sir. Yes, it is available. Yes. Yes, it is visible now.
The screen is, yeah. Of my today is 2D cell culture systems for natural molecule drug development and delivery systems. Transition to 3D, focus on certain hallmarks of cancer. I've been told by the organizers they have about an hour. So, so first of all, I want to give you a reason for why work on natural products. Because half of all the anti-cancer drugs approved internationally are either natural products or the derivatives, and on the basis of knowledge gained from small molecules or macromolecules that exist in nature. Okay. And under the natural products category, we are focusing on flavonoids. They're an important source of anti-cancer drugs. But 60% of the polyphenols in the diet are flavonoids. Okay, they have this fundamental structure here, A and B benzene rings and oxygenated heterocycle in between. On the flavonoids, we are actually focusing on a molecule called luteolin. Okay, in case the slide is not visible. Okay, so you find this luteolin is present in onions. We have quercetin present in beans. So there are a lot of food items which have these flavonoids. See, one of the problems with working with flavonoids is the importance of bioavailability. which means a lot of times these molecules don't reach site of action in sufficient quantities. And so you need to. So basically, the outline deals with the ligand, that is luteolin. Then I'll be discussing about the target and then the model. So I'm trying to fit in my lecture and contextualize it in the context of the team in terms of 2D to 3D. So once you figured out the ligand, just have to look at the model. Just going to step back a little and show you the general mixed audience. And
Sir, sorry to disturb you. Your voice is breaking.
by the way, I'll be taking the questions. Is breaking, is it?
Yes, sir.
Is it okay now?
It's better now.
Is it clear now?
Yeah, it's clear now.
Aha. Oh, okay, fine. Let's step back a little, and I'm projecting the general structure of the epidermis. I am told the audience is diverse. There's some students and some senior researchers. For the senior researchers, this may appear rudimentary, but I'm catering to the students who are tuned for join.
Participant, please bear with us. I think he have lost his connection. He'll be joining soon, like just in a few seconds. Just give it a minute or so. I'll just ask him.
Hello, can you hear me?
Yeah. Yes, we can hear you now.
Okay. Share screen. Okay, I think there are going to be some connectivity issues. At least let me send you the message that I have. So the idea here, we are trying to trigger a terminal differentiation like cell death in our model systems. And this picture is relating quantification and apoptosis. In the case of apoptosis, you know, there is chromatin condensation, cell membrane blebbing, formation of apoptotic bodies, right, and the degradation of DNA, all right. So this will give you a broad overview of the extrinsic and intrinsic pathways of cell death. So what am I getting at? I identify luteolin as the model flavonoid, identified the skin as a possible tissue, and then we talk about cell death because when we talk about cancer, one of the hallmark features of cancer is resisting cell death, and you want to selectively induce cell death in cancer cells without harming the neighboring cells. But do we take the epidermis into account, or do we start off with 2D cultures and then work our way up to higher order, more reliable and relevant model systems? Okay. So I just want to tell you that the unifying theme involves caspase 3 with extrinsic and intrinsic, and you can trigger cell death. Now, we are not going to talk about a target. The protein is called caspase 14. Protein is caspase 14. It's involved in terminal differentiation, okay, and expression is restricted to the cornifying layers, okay, epidermal terminal differentiation. And they've also found out that the 14 in the stratum corneum is the one that is actually active. How did they find out? The immunodepleted caspase-14, the activity fell down. So came down. So when you talk about enzyme for the students, you think about a substrate and convert it to product. Okay, and this is a zymogen that needs to be proteolytically activated to perform its function. Okay, and then so you may wonder what the function is. It has one function, is to convert this filaggrin. So you may wonder where this filaggrin is localized. You go back to this slide here. Here you see filaggrin, right? Right here, filaggrin. So profilaggrin is converted. Filaggrin is a natural moisturizer that is present in our skin. All right. So basically, either the caspase-14 breaking down filaggrin fragments, or it inactivates an inhibitor, activates an exoendopeptidase, is involved in breaking down filaggrin into amino acid. It acts like a natural moisturizer, right? Apart from that, caspase-14 is actually degrading the inhibitor of the caspase-active DNase. Caspase-active DNA, it gets to the nucleus and degrades DNA. And that's one feature that's in common with cell death and this whole terminal differentiation, DNA degradation. You also have another pathway involving enterokinase and mesotrypsin. Okay, now what I'm doing here, I'm trying to give you a conceptual understanding of what we have done thus far in terms of identifying a ligand, identifying a target. And now we're going to go back and ask the question, can this flavonoid, model flavonoid, induce cell death in a human keratinocyte cell line? So we have at our disposal the HARCAT cells, right? HARCAT cells that have been spontaneously immortalized. This is the original paper documenting that. And then the advantage with this with this HARCAT is that you can modulate the calcium level and induce differentiation. All right. So it's a good model wherein you tinker or modulate the calcium levels and induce a differentiated phenotype. And how do you know it is differentiated? Obviously, you look for differentiation associated markers like keratin 1, keratin 10, and involucrin, right? So I think this is an important slide. Why select HARCAT in the first place? It can exhibit this morphogenesis and differentiation. It can also have get stratified. It can go back and forth between differentiated and basal state upon changing the calcium concentration. And you know it's differentiated based on the expression of some key differentiation markers. Okay, so immortalized cell line, from our point of view, we want to demonstrate a proof of concept by hello, can you hear me?
Yeah, now we can hear you.
Yeah, we are I'm extremely sorry. This I didn't anticipate that the internet would not work. No problem. So this is the protocol, and then you can do different experiments. You can do Western blotting to look for differentiation markers. You can look at the proliferation. You see it's more proliferative under low calcium conditions. You can also do immunofluorescence to demonstrate the presence of these markers, right? Western more molecular immunofluorescence, cytological method. Now, I've run through a few slides just to save on time, not for any other reason, as to why we selected luteolin. Because in our hands, we found the IC50 value was good, and this is actually work done by 𝐊𝐨𝐬𝐬𝐿 et al. In 2008. It can cause cell cycle arrest, it can cause apoptosis. Remember, I just showed you a picture very briefly about intrinsic and extrinsic pathways. And you have an IC50 value, that's a variation. So that's one of the issues pertaining to our work, wherein we do see variability depending on our defined experimental setup, the protocol, and the need to validate the protocol across labs in order to get comparable numbers. All right. But the fact it has been demonstrated that the flavonoid luteolin has anti-carcinogenic effects. This is actually our work published some time back, quite some time back, wherein luteolin has shown cytotoxicity. Again, we had this work repeated a couple of times. So this is one of our doctoral students who has finished. And then the next doctoral student carried on and repeat the work. There's some variation in the numbers. All the more the reason why we need to validate our protocols across laboratories. Nevertheless, with the relevance of the control, the reference to the control, it exhibited cytotoxicity, also in apoptosis, inhibited colony formation, and then cell cycle arrest, G2M arrest. Okay, so this was the first work wherein we actually demonstrated that luteolin was able to induce cell death in the HARCAD cells, and then we carried on that work, and now we're now linking. So I hope, in spite of the connectivity issues, the message, the essence is clear, that we have a ligand, we have a target, and then we have a model, 2D model of HARCAD cells. So this is what it is. So this slide is an important one because we're linking the ligand, the target, and the model. And we find, with reference to the positive control, in this case, the positive control is vitamin D3. Okay. So it's able to induce caspase-14 expression. So all of you know there are different ways of measuring a protein. You can do a Western, you can do ELISA, or you can do activity-based assays wherein the substrate is converted to product. Okay, so functional assays are usually preferred. So we also looked at the RT-PCR caspase-14 expression, reference to the reference value, and this slide actually tells you that luteolin is doing better in terms of caspase-14 expression, right? And then we also looked at involucrin. You know, some of you may know involucrin is a marker for differentiation. So if you have caspase-14 activity and concomitant expression of involucrin, it's an indirect way to know that there's a differentiating environment, right? Now, earlier, I do not know if you remember, I said one of the limitations of these flavonoids is the issue of bioavailability. So 2014, there was a paper on luteolin in chemoprevention, in vitro and vivo anti-cancer activity. They used nano-luteolin, and they found that they were able to improve the IC50 value. So again, at the two, they tried lung cancer cells and squamous cell carcinoma head and neck. So this is our, I should say, fairly groundbreaking work performed by one of our PhD scholars, internal part-time scholar, faculty, or since left and joined the industry. Basically, we encapsulated luteolin in liposomes, pegylated liposomes, and we found an increase in caspase-3 activity. You may recall that the unifying theme in the apoptosis diagram is the caspase-3 activation. So this demonstrates fairly clearly the improvements in caspase-3 activity, showing that if you deliver it using this method, bioavailability issues can you hear me?
Yes, sir.
Yes, we are shifting gears here a little bit, and I'm sure Advances is currently involved in 3D models. Basically, the overview, conceptually, without getting into too much of the methodological details, you are creating a proliferating environment in the submerged culture, and then you're shifting it into air-liquid interface, cell insert system, cell culture insert type of setup. And then when you're exposed to the air, that's where the cornification actually starts, the differentiation. So that is the overview. And this is actually work published some time back, wherein they compared the reconstituted human epidermis with the normal epidermis, and then as before, they look for different markers, and they were able to detect those markers, right? Reconstituted human epidermis. Now, again, to bring us back to the issue of caspase-14, and this fits in very nicely with the theme of the conference, because there is very clear-cut indication here to show that in a 3D model. Here, they have purchased the 3D models from Mattek Corporation, and they have shown that the caspase-14 expression is different if you were to do a 2D versus a 3D, and also in response to dexamethasone, which says dex increases caspase-14 in both 3D skin models. And you see here, in the case of 2D, it actually decreases expression in undifferentiated, but not in differentiated. So that depending on the stage of expression, differentiation of these cells, caspase-14 expression is going to alter. So it's all the more the reason for us to actually shift to these 3D models to better get a better handle on the IC50 value, specifically for luteolin. But it would be good to develop the stratified structure starting from Harket cells. Okay, so this is actually the graphical form. You see caspase-14 activity better, AP200 model, and DEX is able to increase caspase-14 expression, right? So this is looked at the gene level and also at the protein level. See, one of the things that we noticed in another paper is they were able to go to much smaller concentrations of vitamin D3 and induce 14 expression. So this is another area where there's possible hormetic effects we need to refine our dose range to see what kind of results we get. All right. Okay, so just to recap, I gave you a reason of why we want to work on natural products as an anti-cancer agent, and the rationale for picking luteolin, and then give you a rationale for inducing caspase-14, hopefully a terminal differentiation mediated cell death. And I gave you a reason for picking HARCAT cells, because those that model, you can easily modulate the calcium levels and make it differentiate. And so that would be the next goal. And the fact that in the case of tumor, see, all along we're talking about normal epidermis and stratification, things like that. But actually, we are interested in tumor-associated alteration, which is why we picked up the HARCAT cell, the immortal cell line. The interesting thing is, depending on the cancer, the caspase-14 expression is altered. For example, caspase-14 expression is increased in ductal carcinoma in situ and invasive cancers of the mammary gland. So again, you have to be very careful with the choice of the model, the choice of the target, because the target is going to behave differently in different types of cancers. All right. So it's a very intriguing enzyme. Okay. We're just shifting gears a little bit. All along, we talked about dermal systems. Now, if we're shifting to breast cancer. Dr. Kadam, are you able to hear me?
Yes, sir. Yeah.
Good. Okay. So we are now moving, because of connectivity issues, I'm trying to summarize so that you get the essence. And I think Dr. Sachin can share the PPT with the participants, no issues. So I'm providing a reason of picking MCF7, MD, MB31. There is a reason because in terms of the molecular classification, in certain two separate categories, these cell lines would be represented. Obviously, there are other issues which we'll get to. This is our work published some time back in terms of against caspase-3, migration, maturation, and migration inhibition. So one of the features of the invasive phenotype is migration. If you inhibit migration at the 2D level, you have established proof of concept at that level. So we did some extracts apart from pure synthesized chemical derivatives of flavonoids like luteolin. We also worked on extracts, and we find there is inhibition of migration and procaspase-3 maturation. Okay. So just skip this slide. And apart from the IMODI, we have also looked at oroxylum indicum, and here is MDA MB231 cancer cells. We demonstrate cytotoxicity, apoptosis induction, anti-metastatic potential. Okay, now so much for our work. We all know that there are stem cells, cancer stem cells, even though there are some who believe in the stochastic model. The stochastic model basically says some cells are more resistant than others. There are those who believe in stem cells actually say they are cancer stem cells. So if you want to develop a good drug, you would want to target these radio-resistant and chemo-resistant cancer stem cells. Okay. So in terms of functional classification, you have these stem cells, and so this is a good reason to target this subset of cancer cells. They can be transformed by various oncogenes to give rise to cancer, be it the stem cell or the progenitor cell. So this is a good overview picture. You have stem cells which are resistant to chemotherapy and radiotherapy. Okay, drug resistance. So a lot of times a person will have cancer, he or she will be treated, and the tumor grows back. And it's believed that these resistant stem cells, which have evaded chemo and or radiotherapy, they grow back. The other hallmark of cancer is spreading of the cancer, primary or the secondary site, or reseeding the primary site. Metastasis, wherein an EMT program is involved. And what is the EMT? It's actually the epithelial mesenchymal transition. You know, the cancer stem cells also exhibit this mesenchymal stem cell properties. Okay, so when you look at these breast cancer stem cells, obviously from the methodological point of view, if you are going to enrich for these stem cells, you can either start from the cell lines or you can start from explants and then grow them and then enrich for the breast cancer stem cell. We look for surface markers. One important marker, the CD44 plus and CD24 minus. Okay. All right. Okay, you can try it in 2D and also in 3D, and and also think about the transition of these stem cells between epithelial and mesenchymal. There is plasticity. Okay, so that is an important point to keep in mind. You have to get rid of the cancer stem cells. You also have to get rid of the other cells which are more resistant. If you don't believe in the stem cell, you say more resistant, less resistant. You need to know whether it's epithelial, mesenchymal, and you want to selectively eliminate those cells. And then if you want to link the EMT transition with epigenetics, okay, I just want to show you this picture. It's actually published by Tam and Weinberg. And why is this so very important? Because I said cancer stem cells do exhibit EMT, and those cells which get dislodged from the primary tumor and intravarsate into the blood vessel, they also exhibit this EMT process. So in terms of at least a couple of hallmarks, EMT transitions are quite important. And these EMT transitions, which you see here, change in shape, and some molecular markers, EMT transcription factors, upregulated, snail, twist, slug, zeb, ZEB1, so on and so forth. Loss of E-cadherin, upregulation, they can be correlated to epigenetic changes. And this is, as an analogy, let's say you're going by road from one city to the next. If you know the intermediate stops and you see them, you know you're heading the right direction. Otherwise, you don't see those stops, you know you're heading the wrong direction. Similarly, if you're able to characterize these intermediate states in terms of the molecular aspects, especially epigenetic features, you will be able to find out the mechanisms involved in the transition of the epithelial-like cell to mesenchymal cell, and thereby you can look for druggable targets, right? So again, to contextualize my talk with reference to what we are doing, again, you see luteolin has been reported to suppress metastasis by reversing EMT by downregulation of beta-catenin. So again, this whole idea of taking the cell lines, converting in 3D on one hand, on the other hand, enriching for stem cells, and looking at the EMT features and see if you can reverse EMT and then improve your delivery, decrease the IC50 in a 3D system, right? So one of the examples is it's not so much the small molecule inhibitor, the fact that this small molecule inhibitor is working as efficiently as luteolin to eradicate the cancer stem cell population. So I think we are heading in the right direction. Another molecule that we're working on is pterostilbene. It's involved. It needs an estrogen receptor. So if you have a cell line that does not have estrogen receptor, could you make it re-express and they may act as a sensitizing agent for other molecules to be used as part of a combination therapy to get rid of these breast cancer cells, right? Okay. Okay, we're back on. Yeah, this connectivity issue is a little bit unsettling, but nevertheless, I hope I have driven home the message of the limitations of working with 2D. So when you think about spheroids, it's more representative of the tumor mass that you see in vivo. Obviously, there are other issues. Obviously, you need to include the vascular component, the avascular, but then you have a gradient in terms of oxygen, nutrients, ATP, and so on and so forth. The inner area where it's necrotic, okay, and then there's a quiescent zone, and this area is proliferating. So conceptually, if you want to have, if you have a drug combination, one which will attack the proliferating cells and one which attacks the cells here, then you will be able to get rid of the tumor mass. And so this model is a good one, a better one than the 2D cultures, multi-layered spheroid. You can also do a hanging drop. Basically, you take the cells, trypsinize it, count it, put it in form of drops on the cover of a petri plate, invert it back on, and wait for some time so that the aggregate forms, right? You can also use laminin-rich ECM, which represents the basal membrane, or you could use scaffold. So by and large, in terms of stratification, it's both scaffold but scaffold-free. I'm sure Advanced Cells are doing a lot of advanced research into higher order model systems, either using a scaffold or scaffold-free. Other way to classify is stagnant or something that is not. You know, if you want to have better mass transfer, you would use a dynamic system like a rotary flask or a spinner flask, right? This is evidence to show that this 3D model is a good one. Obviously, there are issues of reproducibility in terms of size, shape, volume, density. Okay, and you need to get protocol that make it reproducible. You find here, if I look at hypoxia here. Hypoxia was observed only in the dense multicellular spheroid. So, some formed loose spheroids, MCF7, MDA, MB, but other cell lines which form dense. The dense ones are more resistant to these drugs. So when you are using this as a model, you want to develop a model that is relevant to the in vivo situation. So you may want to pick on the dense multicellular spheroids. Hypoxia, then you look at the proliferation markers, and so you find a dormant subpopulation in 3D that is contributing to the drug resistance, the quiescent population. So all of this is actually giving us a reason for moving away from 2D and going to 3D models. Okay, just skip these slides. So another paper where they reiterate the importance of size-induced tumor progression. Some of these triple negative cell lines did not show size-dependent upregulation of mesenchymal markers. Okay. So when you're developing a model that should be clinically relevant, you should use the appropriate model for you to replicate what is seen in vivo, right? So you find your triple negative cell line do not exhibit size-dependent upregulation of mesenchymal markers. Okay. So apart from cell death, which is one of the hallmark features, and the other hallmark is cellular EMT, seen in the case of stem cells, and also for introversation. So the question is, what is introversation? When you have a primary tumor, in certain conditions, it loses its cell-cell connectivity, connections, and it gets dislodged and invades via the basement membrane and gets into the blood vessel. And you can model that introversation. You do a 3D co-cultivation of MCF7 cells and the lymphoendothelial cells. Okay, a model for this introversation and take it to the spheroid state, right? So you develop the MCF7 spheroids first. Obviously, you prepare cell suspension, you do a cell count, and then you use an appropriate low cell attachment plate, and then you transfer it to ADC monolayer, right? Obviously, when you talk about tumors, we need to take into account all the cells. So the higher order model system development becomes all the more challenging. Remember, I told you spheroids may not take into account the vascular part, so you need to incorporate okay. I hope I'm audible. Can take it forward. to a xenograft model, wherein get these cells and implant it either ectopically or orthotopically, and then see if your drug works. In vivo context, you can also develop genetically engineered animals, and then patient-derived xenograft. So the context, the theme of today's symposium, cell lines, cell line-derived xenograft, patient-derived xenograft. Okay. In terms of spheroids, multicellular tumor spheroids with some cell lines, multicellular tumor-derived spheroids, which would be from the tissue sample taken, and then you can do heterotypic culturing, more than one cell type. So this is a good overview representation, pictorial representation. Just goes to show that if you want to resemble in vivo response, you got to move away from these monolayers and go more towards patient-derived xenografts. Obviously, there are some, you have bioprinting and organ on a chip somewhere in between. I'm sure the other speakers will be talking about it more in greater detail. And so here, see a picture here, linking animals and humans. If you were to go with a standard therapy, you find the therapy is not always optimal. So what can be done is blood is collected for copy number analysis, alteration analysis, and then you take the tumor fragments. Obviously, informed consent is important. Ethical clearance is important. It is xenografted into mice to develop a patient-derived xenograft model. So now you have the patient tumor, you have the patient-derived xenograft sample, you have blood, you do a comparison of the genomic and gene expression analysis. Then since you have an animal model, you can treat it with certain drugs, chemicals, biopharmaceutical antibodies, small molecules, based on the genetic analysis, initial analysis, and finally, after treatment, you can identify those tumors that are resistant to treatment. So if relapse occurs, you take another biopsy, analyze the tumor which recurred and also the tumor that was refractory to treatment in the animal model. Again, you can compare. And that way you can go through this personalized medicine strategy, integrating data from the human, take the explant and put it, graft it. Tumor sample grafted on an animal. You have genetic characterization of sample from the blood and the tumor and the xenograft model. And you compare before and after treatment, the humans as well as the animals, and that way you have a better idea of what would be the expression profile in those tumor subsets that were refractory to treatment, and can you actually modify the treatment strategy and make it better. This is actually my last slide. I want to thank our research scholars for having contributed. In fact, the Caspase-14 work with encapsulation luteolin was done by 𝐕𝐭, who has since left. Yeah, I want to, can you hear me?
Yes, yes, we can hear you.
Yeah, I want to thank the organizer once again, and I want to thank the participants for their patience. I want to apologize to both for this issue with the internet, but I hope I was able to convey the essence of what I am getting at in terms of our work with HARCAT and MCF7 and MB231, and where we hope to head towards, especially in the context of caspase 14, especially since the expression is altered depending on the stage of differentiation. And that can change, be it 2D or 3D, and how we can refine our IC50 values so that it is more relevant to the in vivo situation in terms of developing drugs from natural molecules, anti-cancer drugs from natural molecules. Thank you.
Thank you, Professor Suresh.